All right, good morning, everyone, or good afternoon, depending on where you are joining us today from. Before I introduce our speaker for today, I would like to remind everyone if you have any questions during the seminar, please send your questions through the Q&A, preferably, but you can also comment through the chat. All right, with that, it is my pleasure today to introduce Dr. Scott Gray-Owen, professor at the Department of Molecular Genetics at the University of Toronto. Dr. Gray-Owen is a microbiologist, cell biologist, and immunologist with a particular focus on understanding molecular and immunologic interactions between humans and disease-causing bacterial and viral pathogens. Dr. Gray-Owen's overarching goal is to understand how these microbes adapt to avoid what are otherwise effective immune defense mechanisms and to differentiate between pathogenic and protective immune responses so that effective interventions can be developed to avoid pathogenic responses while harnessing protective responses. Dr. Gray-Owen has many accomplishments. Beyond leading his own research group's efforts, Dr. Gray-Owen is the director of the Toronto High Containment Laboratories, which includes Molecular and Immunology Core, as well as the Biosafety Containment Level 3 facilities to support academic, government, and industrial infectious disease research. Dr. Gray-Owen is also the director of the Emerging and Pathogenic Infections Consortium, which supports the broad research community's ongoing focus on combating the highest priority infectious challenges. And Dr. Gray-Owen is the co-director of the Canadian Hub for Health Intelligence and Innovation in Infectious Diseases, which brings together Ontario's academic institutions, government agencies, and industry partners to promote pandemic preparedness, sustainability, and resilience in Canada's biomanufacturing industry. Dr. Gray-Owen is also co-founder and chief operating officer of Engineered Antigens, Inc., a company focused on structure-based design of vaccine immunogens targeting human and livestock pathogens. On behalf of IUSTI Canada, I welcome Dr. Gray-Owen.
Thank you, Samudu, and thanks for the opportunity to be here. It's really a pleasure for me to get the opportunity to talk about something that I consider so biologically interesting and both on a clinical and public health level, so very important. So I'm going to start just with a brief overview of the plan summary for today, and then pass through this, and I'll gauge my talk today, the length of my talk today, based on where I want to go. There's so many interesting things that have happened in vaccines and understanding of gonococcal infection over the years that I tend to extend on the background and get engaged in it. So I'll keep an eye on where we're going today. So I want to start with a little bit of background on infection and immunity with Neisseria gonorrhea and its closely related cousin, Neisseria meningitidis, because it's important for where we are with vaccines today. I'll talk about the gonococcal vaccines that have been assessed and where we are right now. A little bit of work that we've done on trying to understand the correlates of protection so that we could try and understand what we might be looking for, what types of immune response we might be wanting to target into the development of vaccines. And then if I have time today, I'm going to talk about the work that my group has been doing, trying to look at a critical pathway for infection, that's iron acquisition by the bacteria, and our attempts to target this with vaccines. And related to the last point, and Samudu mentioned it, I'm co-founder and chief scientific officer of Engineered Antigens, Inc., which is focused on the protein structure-based engineering of vaccine immunogens that target human and livestock pathogens, including Neisseria gonorrhea. And so it's important to disclose up front today. Okay, so the pathogenic Neisseria, I don't want to spend a lot of time talking about background with Neisseria gonorrhea today, but obviously this is a really important pathogen, both by virtue of the number of cases that occur each year and the potential, the impact of it, both acutely and the sequelae that pass beyond that. the disease itself is really not the bacteria causing any overt damage to the tissues. The gonococci aren't themselves typically cytotoxic or anything else, but really what they are is, I think of them as an elegant pathogen that is able to get into the tissues and go undetected. And by the time they are detected, there's a lot of them there, and we get this massive inflammatory response that causes the tissue damage. And the damage then occurs wherever the bacteria might be. Acquired blindness if it's in the eye, pelvic inflammatory disease, and female infertility if it's in the female upper genital tract, for example. And I'll touch on today, part of the problem that we have with the gonococci is that we have very little adaptive immune response or memory to infection. So individuals get infected over and over again. And this is important for a sexually transmitted infection, as they tend to circulate within high-risk groups and then move out into other populations beyond that. And an interest in my group and many's, and I think a public health importance, is the fact that gonococcal infection, despite the fact that it's not overtly damaging the tissues or causing, ulcers or other types of things that might make it obvious why it would promote HIV transmission, it causes individuals that have HIV to shed more virus, and it causes people who have gonorrhea that are exposed to HIV more susceptible to infection. And so it has a really impact on the epidemiology of HIV. Meningococci, as I said, it's closely related. The biggest difference between them is that the Neisseria meningitidis has a polysaccharide capsule that encompasses it, and this allows it to survive in the air as it moves from one individual to another. It's much more rare if we think of disease, but it's actually quite a prevalent bacteria in that it's in the throats of many healthy individuals. And obviously, the disease itself is also inflammatory. It's also an over-response of the innate immune system, or the inflammatory system, to the bacteria. But in contrast to the outcomes and mucosal infections, primarily with the gonococci, the rapidly progressing invasive disease by Neisseria meningitidis is often fatal. And so this is a real problem. And I touch on both of these because of the fact that, as we all know, the meningococcal vaccine really has opened our eyes to the potential for a gonococcal vaccine. So my group has studied both the meningococci and the gonococci for several decades now. And for most of that time, people said, meningococci is so scary and important. Why do you study gonorrhea? And my interest was often that it's important, and it was biologically and immunologically a very interesting pathogen by virtue of how it causes infection and persists within mucosal tissues. This is completely switched now. We have meningococcal vaccines, and we have really an increasing burden of gonococcal infection. And as we all know, there's an increase in antimicrobial resistance in Neisseria gonorrhea. Every antibiotic that tends to be used clinically for this bacteria, the bacteria become resistant. And the bacteria acquires this resistance and maintains it. And so we can't go back and use antibiotics in most cases that were used in the past because resistant remains in the population. And so this has really led to the potential fears and in some cases actual instances of what are effectively untreatable Neisseria gonorrhea. So this has really become the priority, and global health, public health agencies and others have really made it clear that this is a priority to target this pathogen. It's a real concern. So regardless of whether we're talking about Neisseria meningitidis in the throat or Neisseria gonorrhea on the genital or pharyngeal mucosal tissues, infection effectively looks like this. The bacteria reach out and touch the apical surface of the mucosal epithelia using their pilus, and this retracts to pull it down and allows a engagement of the outer membrane proteins, adhesins, known as the OPA proteins, being one example, to their host cellular receptors on the surface of the epithelial cells. And this allows the bacteria to penetrate into the cell and actually pass through the epithelial cell. Often, at least in vitro studies, this doesn't break down the epithelial barrier. The bacteria are able to pass through and exit into the subepithelial space. And thinking about it this way, there's really three important points to consider here as we're trying to tackle this bacteria. First of all, the bacteria live intimately associated with the mucosal epithelial or even in the subepithelial space. And therefore, its entire lifestyle is about avoiding the innate and adaptive immune system. And it really does a remarkable job at this. It can actively suppress adaptive immune responses, and it can vary its surface at a very high rate. And this is both a differentiation of antigens at the surface, sequence variants between different strains of gonococci, but even within an individual strain, there can be recombination that goes on between various expressed and silent alleles that can either make a new antigenic variant or actually, in the case of phase variability, turn on and off antigens. And when an immune response targets an antigen, then it tends to select for a variant, for example, in the tissue that might turn that off if the protein's not essential. So this makes it really difficult. I said a tough nut to crack. This makes it a really difficult bacteria to target. The other important thing from here is the fact that these bacteria, buying everything that they touch, is specific for their host. So the only niche for both the pathogenic Neisseria is within humans, and the gonococci, it's mostly within the genital tract, although it can obviously infect the pharynx and the rectum as well. And so by virtue of the fact that it only lives within humans, it can become very exquisitely adapted to life here. And so it doesn't make amino acids that are available to it in the tissues, for example. And it can bind to host proteins that evolve to prohibit other bacteria, just general bacteria, from binding to them, including these CCAM receptors, which are on the surface of the epithelial cell. Neisseria will only bind to the human version of these, for example. And so this prevents us from having an animal model to study infection. It prevents us from having an animal model to develop therapies and vaccines against it. And this is something important I'll come back to later on. And so,
sorry, I'm having trouble hearing you.
As I said, sorry, Siri is answering here. As I said, we don't get an adaptive response to infection. And perhaps the best example of that, the most clear example of that, is a paper that's now 25 or so years old from Mike Russell's group. And Mike was a real hardcore mucosal immunologist who was interested in infections in the genital tract, and particularly Neisseria gonorrhea. And in this paper, as it said, they monitored individuals who had gonorrhea and looked at both the mucosal and systemic response to the gonococci, and in both men and women. And as the underline what I've underlined here, in general, the local and systemic immune responses to the gonococci were extremely modest. And on the right-hand side, you can see actually the cervical mucus in women, and looking at IgA, IgA1, IgA2, and IgG in the cervical mucus, and really low levels in each case. But more important than that, or as important in that, I don't know if you can see at the bottom, in black is where the individuals who had current infection. In white, they don't have infection. And the first two are individuals who don't have a history of gonorrhea before, and the second two are individuals who have had gonorrhea before. And effectively, all I want to show you here is that there's no difference in the antibody, the anti-gonococcal antibody in any of these. So you don't see a increase in antibodies to primary infection. In fact, others, Mike and others, have shown there might be a modest increase in IgM that's transient and goes away and never converts to IgA or IgG with infection. But then when we get infected again, when individuals get infected again, there's no memory to this. There's no boosting of it. And so this has made it really difficult to understand what confers protection against the gonococci. So obviously, there's been a longstanding interest in developing a vaccines that would protect against gonococcal infections. But in many cases, individuals said, well, natural infection doesn't confer immunity, So how could a vaccine do the same? And clearly, I think the answer to that is that we prepare vaccines to elicit a response against antigens that the bacteria have learned to avoid immune responses against. And so we want to package this and provide this in a way so we can recognize infection different than what natural infection did. However, a number of classical studies have tried to immunize against the gonococci and really shown that they've really been unsuccessful. And this is a table that I took from a recent really excellent review article on this. So whether they used heat-killed, partially lysed gonococci, and this was actually in part a study done in Canada in Inuvik, Pillin subunits against the homologous strains or heterologous strains, or preparations of outer membrane antigens. And this was targeting the porin, the immunodominant antigen actually, but was contaminated with other proteins as well. In most cases, there's absolutely no evidence of protection, and that includes when they do the trial in high-risk individuals, in general, high-risk military individuals, or controlled human infection model challenges. So where they're infecting individuals experimentally, urethral infection of men after immunization. There was some level of protection against the homologous strain. So if they immunize with a Pillin subunit and then express the same strain, the same isolate that expresses that Pillin, you can get modest protection, but not protection against other heterologous strains. And so the most recent of these, the one at the bottom, actually was published more than 30 years ago. And for much of this time, there has been really a fear that these types of data suggest that we couldn't get vaccine-mediated immunity to it. And I think a real paradigm shift, something that shook up the field really was this Lancet paper in 2017 by Helen Petousis-Harris, where this was effectively took advantage of the fact that there was a meningococcal epidemic in New Zealand, and they Chiron at the time produced an outer membrane vesicle vaccine targeting this one meningococcal strain. And this conferred protection, and so children and both young children and children up to the age of 20 were immunized with this, and it helped eradicate this epidemic. And so this was a real big success story for the meningococcal vaccine itself, this MEN-ZB, outer membrane vesicle-based vaccine. So this group took advantage of the fact that as these individuals aged, they could look at the different rates of gonococcal infection in those that the cohort that had received the vaccine, which was most of the kids in New Zealand, and compare rates of gonorrhea versus chlamydia in these versus others. And their conclusion, shown here in the abstract, but shown also on the right-hand side, was that there was a 31% reduction in infection of the individuals who had been immunized with the Group B meningococcal outer membrane vesicle vaccine, this MENS-ZB vaccine. And so I couldn't help but put this in. This is from Dumb and Dumber, and he's saying, yeah, so you're telling me there's a chance. Yeah. So 31% isn't huge, but it can clearly have an impact when we consider that the fact that there are 90 million cases of gonorrhea in the world. And so we really have to understand what the potential impact of this vaccine might be and whether it's worthwhile rolling as it's rolled out to target meningococcal disease, whether it will have an impact on carriage of gonococci as well, or whether we can use this strategy to produce a vaccine that targets the gonococci. And so this led to a World Health Organization meeting to come up with the preferred product characteristics for a gonococcal vaccine. And this is on the right is from the executive summary. And so really there was 3 points that they drove home. This vaccine should be something that prevents gonococcal infection. So it prevents colonization of the genital tract or the throat or the rectum against gonococci. This wouldn't be a good thing if it only reduced disease and didn't reduce that either the absolute colonization of the mucosa or the duration of infection of these, because we'd end up with many more asymptomatic individuals that would have the potential to spread the infection. Really important. The preferred target populations may vary. So in some places, it might be all young people. In others, it might be used to target high-risk individuals, men who have sex with men, sex workers, or others. And so really, this would be determined once we have an effective vaccine, then we could figure out what's most cost-effective and what could be acceptable to the public as far as the implementation of this vaccine. And the other kind of asterisk here is that we have to consider that we might get a vaccine that works against some types of infection and not others. So it might be more effective in women or might be more effective in men. And so there would have to be a decision made on the impact of that and the rollout of that. And then related to that is whether we should target everybody or just those at high risk. So how much should this be rolled out on a broad level? What's most cost effective? How we can target the transmission of gonorrhea? And since the gonococci it's only niches within humans, we actually have, with an effective vaccine, a potential to eradicate this infection. So the men's-EB vaccine really was this outer membrane vesicle from the strain NZ-98-254. So this is the New Zealand epidemic strain. This is a really complex antigen. There's about 100 proteins, and it has LOS. And even though they used a detergent to extract lipopolysaccharide or the endotoxin from this, it still is contaminated with the endotoxin. So they are inflammatory on their own. And it's recently been shown by David Stevens that in some cases, even though the lipo-oligosaccharide, the sugars on the surface of the bacteria, which are also on the surface of the outer membrane vesicle, are highly variable between strains. If the gonococcal strain happens to share sugars to this NZ-98-254 when they produce the vaccine, then that can contribute to serum bactericidal activity. So this is a lot of really complex antigen with the potential to provide protection against gonococci via dozens, probably, of different proteins on the bacterial surface or the LOS. Another vaccine that's been rolled out by Pfizer is this Trumemba vaccine, and this was targeting the factor H binding protein. And this is the protein that allows Neisseria, the Neisseria meningitidis, to evade killing by the complement system. And so they picked two different variants of this, and that's what the meningococcal vaccine from Pfizer is comprised of. The GSK serogroup B vaccine, 4C-MenB, or marketed in many places around the world as Bexsero, including in Canada, is actually a 4-component vaccine on its simplest sense. It has one factor H binding protein. It has Neisseria adhesin A, Neisseria heparin binding antigen, NHBA, and it has the same outer membrane vesicle from the strain that was originally used in New Zealand. The original pertussis-Harris study was done with the men's ZB vaccine. Thought that the Bexsero might have the potential to do similar or even better than that. What's important, though, is that in gonococci, FHBP is probably not relevant in protection, and that this protein is curiously expressed only in the cytosol of Neisseria gonorrhoeae. This doesn't make a lot of sense if the only function of it is as binding factor H, but that's where it is. NAD-A is completely absent in Neisseria gonorrhoeae, and gonococci does share NHBA. It's about 60% similar, but I believe it's only in about a third of the strain, so this can't confer all the protection or broad protection against all the isolates. And so it's a combination of these two antigens that have the potential to contribute to infection. What's important is that these vaccines, both Trumemba, men's-EB, and Bexsero, have proven effective against reducing invasive disease by meningococci, and this was their primary goal. What's been somewhat disappointing is that they do not reduce pharyngeal carriage of the meningococci in the population. And so individuals who have these vaccines should have a significantly reduced chance of getting invasive disease by serogroup B meningococci, but they'll still carry it in their throat. And as I said, this is a bit of a challenge because now that means we can't eradicate it, and it means there's individuals, the vaccinated individuals could still spread the infection. This makes it hard to understand how a vaccine like this could protect against gonococcal infection. A different species of bacteria, so they're more heterologous than the meningococci, and obviously in a different site. However, Helen Marshall's group in Adelaide in Australia took advantage of the fact that 4C-MenB is used broadly in Australia. There's immunization program for infants, children, adolescents, and young people in South Australia. And this was a study where they were looking at the vaccine effectiveness of 4C-MenB primarily against meningococcal disease, but they also looked at rates of gonorrhea within this. And vaccine efficacy was really high in individuals. In fact, there was very few cases of invasive meningococcal disease by group B strains in the children who are immunized. And so this is a hugely gratifying, this is a huge success. What they noticed, and this aligns pretty much with the study that we talked about previously, is that there was also a reduction in the rates of gonorrhea in adolescents who had been immunized with 4C-MenB when they were younger. And so the efficacy was about 35% up to 3 years, and then when they tested after 3 years, it dropped to about 23%. So it could be several years of some modest protection. And so again, very gratifying that this might have an effect. And I should say, as previously, the control for this was looking at the rates of chlamydia. And so the rates of chlamydia weren't affected, weren't different in those immunized versus not. And so in the UK, their advisory board suggested that 4C-MenB, or the Bexsero vaccine, should be used where individuals were at high risk of gonococcal infection. So understanding that this wasn't perfect protection, it was modest protection. Because of the rates of gonococcal infection, this has the potential to have an impact. And so there's really 3 points here. They said it could be used, it could be implemented for those at great risk. And this wouldn't be rolled out as a broad vaccination campaign. Instead, it would be STD clinics and sexual health services determine when an individual is considered to be of high risk and then offer them the vaccine. And when they're offered the vaccine, it was indicated that there has to be real effective education of that individual. The protection is modest so that they don't think that there's one less sexually transmitted infection that they can have. And so this is, again, really important advisory suggesting that we might be able to have an impact here against the gonococci. GSK produced the 4C-MenB vaccine. They used a process of reverse vaccinology to identify the protein antigens, the recombinant proteins that are used. And then they found the efficacy was much higher when they brought in the outer membrane vesicle vaccine as well. So really, they were aggressive at leaning into this idea of using outer membrane vesicle-based vaccines to try and target the gonococci. If Bexsero could provide about 30% protection, what could we do if we developed a vaccine, an outer membrane vesicle vaccine targeting the gonococci? And so they actually have built recombinant strains that they call these generalized modules for membrane antigens strategy. And this is GMMA or GEMMA. And I think this is cute in that the side part of this is in Latin, GEMMA means 2 things. It means jewel, little jewels, such as these, that might go into the vaccine. And it also means bud, like a plant bud. And so what GEMMA are actually is they make a recombinant strain that breaks the links between the outer membrane and the cell wall, and then overproduce the outer membrane so that the bacteria bleb the outer membrane, these little jewels, the little gemmas. And then they've used a recombinant strain that has a detoxified lipopolysaccharide, so it's less innate inflammatory. Remove antigens that might be distracting, such as RMP, which blocks protective antibody responses against the gonococci. And then they have the ability to overexpress antigens in this that they think would be protective. And so they moved ahead with this very quickly. And in June of 2023, they were given USDA Fast Track designation for investigation to test the efficacy of these vaccines. And they moved into phase 1/2 studies. And really sadly, I think breaking all of our hearts, GSK actually had to stop this vaccine trial because it didn't meet predefined efficacy criteria. So they're completing the immunizations, and they're, I think, really importantly, they're continuing to collect the data, the specimens, the samples, serum samples, mucosal samples, and the data from the individuals who have been immunized. And this will be a wealth of data for all of us to try and understand why this vaccine wasn't protective, or if it had any protective effects in some individuals that aren't seen in the broader vaccine trial. So beyond this GEMMA approach, there's many studies going on right now to test how effective Bexsero really is in different populations. And then there's 3 different companies that are near to entering into phase 1 clinical trials, at least. Intravac in the Netherlands is using a vaccine partially developed by Mike Russell that I mentioned before that includes outer membrane vesicles from gonococci as well as IL-12 microspheres. And in most models, they found that this formulation really promotes a much more cross-protective immune response. So this is interesting. Limitech uses an approach similar to the GEMMA approach, and only that they're using an E. coli-based production system. So they're using E. coli and they're putting gonococcal antigens onto it, carbohydrate and protein antigens onto it, and creating an outer membrane vesicle producing platform. And they believe this allows them to produce the vaccine easier. So that's their strategy. And then Invaxion used an AI-based approach to select potential targets against gonococci, those that were conserved. And I believe Merck now holds the option for this to license this vaccine. So these are those that will go ahead. Beyond this, there's many different groups. I'll just say ourselves, but many different groups around the world who are obviously focused on trying to develop vaccines against the gonococci. I just wanted to take a couple of moments to talk about a paper that we just published a little earlier this year. So with the knowledge that 4C-MenB or Bexsero, the antigens shown in the top right-hand corner, could confer protection against the gonococci. And this is a paper by Kate Sieb in Australia, where they looked at what proteins were shared between the outer membrane vesicle and the recombinant antigens. It says 5 here. There's actually 2 additional ones that are just used as carriers for these proteins, but they're irrelevant in meningococci. And so they found that one of the 5, as I said, NHBA, is shared by gonococci, but many of the proteins, including 21, it's missing here, 21, down below, 21 of the 22 proteins that comprise about 90% of the protein content of the OMV are shared by gonococci. So it could be many different proteins contributing to this. So we use this as a way to try and see if we could understand what confers immunity against the gonococci when in individuals immunized with 4C-MenB. So this is a mouse-based study, and it came from the fact that we noticed that when we immunize animals with Bexsero, we also don't get complete protection. It tended to be about 1/3 to 1/2 of the animals were protected, and the remaining of them were not. And that's shown a little more clearly here. This is looking at each dot is an animal, showing at day by day the bacterial burden within the animals. And in black was immunized with alum, the adjuvant, and in blue was immunized with Bexsero. And as you can see, there is an increase in clearance of the gonococci in some animals, but there remains animals that just don't clear infection. So the blue ones have all been immunized, but there was two groups. There were those that clear more effectively, and there were those that don't clear. And if they didn't clear early on, then they tended not to clear more effectively than the non-immunized animals. And so we thought, okay, great. What's happening in these immunized but non-protected versus these immunized but protected animals? Firstly, I think thinking about how this might be working, looking at the amount of antibody targeting meningococci on the left, the New Zealand strain, or gonococci on the right, this is the strain we used in the mouse infection studies. Clearly, there's a much more antibody targeting the meningococci, so that there is antibody clearly targeting the gonococci, but it's over a log lower than that against meningococci. So if it is about antibody titers targeting, then obviously there would be a difference between meninge and gonococci. So when we look at the this is terminal sera, IgG reacting with the bacteria that we use to infect. Clearly, again, we get a humoral response against the bacteria. This completely does not differ between those that are protected, the yes, the Y down here is yes, and not protected, the no. So it's not just the amount of antibody targeting them. Shown on the right here, it's also not the amount of IgA or immunoglobulin in the vaginal lavages where we're infecting the animals. Really, there's no difference at all between the immunized versus non-immunized, and so we couldn't tease apart those that were protected versus not. We did a great deal of work here looking at lymphocyte populations in the spleen, in the local tissues, and systemically, and saw that they clearly get activated in immunized, but we couldn't differentiate between immunized and protected and non-protected individuals. And really the one thing that we found, and this is looking at each animal, pre-immune serum on the left and immune serum, or sorry, the other way, pre-immune serum on the right and immune serum, and looking at those that differentiate, those antigens that might differentiate between protected and non-protected animals. And really, there were a couple of bands, number 3 and number 6 here, where statistically we can see that the humoral response was higher in those that were protected versus not protected. And this is band 3. Sorry, this is band 6 over here, and this is band 3 right here. There's a typo here. But really, this is not that striking of a difference to us. So it's really difficult to know how meaningful it is. And so it actually might be a combination of responses to different antigens that matter. And so the summary of this study, really a lot of work, was clearly subcutaneous bacterial vaccination can reduce vaginal gonococcal colonization in these animals. The gonococcal specific antibody titers do not differ between protected and non-protected animals. This is both just the titers, the reactivity with the bacteria, and also the serum bactericidal activity against the bacteria. It just doesn't differentiate between determine whether the animal is protected or not protected, as far as we can tell. There's really a robust inflammatory response and adaptive immune response against this vaccine. And in part, probably that's driven by the endotoxin that remains within the vesicles of the vaccine. But again, we looked at broad lymphocyte subsets, T cells versus B cells, but also breaking these down into regulatory cells and Th1 or Th2 subsets of populations. And in no way could we differentiate between the protected and non-protected animals. And if anything, it might be just really more complicated than any of us, even statistically or computationally, can tease out from this. It might not be any one protein or combination of proteins. It might be that different responses to different proteins are what drives protection. So knowing now that we can get protection against the gonococci, at least in some level, we wondered if we selected the appropriate antigen, something that's essential, something that's surface accessible, and something that's strongly immunogenic, could we confer protection against gonococci? And so coming back to the model here, to do this properly, we had to do a couple of different things. As I said, gonococci don't easily colonize anything, don't really colonize anything other than humans and perhaps gorilla and orangutan. That's how evolved they are. We've been using a mouse model to study infection because it has a mucosal tissues, so we can understand the lifestyle of the bacteria within the tissues. And we can bring in the human factors that might support infection to see how these contribute. So what's important is that we needed these human CCAM receptors in the animals. And once it gets into the tissues, we need to provide it with the nutrients that are accessible normally in humans, but not accessible in animals. And so we've been bringing human genes into the mouse and creating more humanized animals. And then we have actually about 11 different human transgenes in the mice in various different combinations. And the impact of that is shown here. And I take the time to mention this because we've had to combine 2 different transgenes for this study. So if we take a wild type mouse shown in the bottom, and this is uninfected, if we infect these with meningococci, this is the nasopharynx, it's true also of the gonococci in the genital tract, the bacteria tend to sit within the mucus, and eventually they'll be washed out within a few days. If we express human CCAM1 on the apical side of the epithelia, which are shown here in red, you can see now the bacteria can tightly associate with the epithelia, and hopefully you can see the green dots, they start to penetrate into the tissues here. And so this allows us to have a colonization, a real intimate association of the bacteria into the tissues. But if we look normally at 7 days, the bacteria are gone. And we thought part of this might be virtue of the fact that it's known that Neisseria can only acquire iron from the tissues of humans or from the blood of humans. And so the iron transport protein in blood, known as human transferrin, if we make mice that express both CCAM and human transferrin, now we can get colonization of the animals out for a week or 10 days until the adaptive response comes in to start to clear them. And so this study is done with a double humanized animals. So I said Neisseria can use iron from human blood, but not from the blood of any other animals. And it can do this because of the fact that it has this really elegant bacterial transferrin receptor system. So this is the outer membrane of the bacteria shown here. That has transferrin binding protein A is a large beta barrel protein that has a plug in the bottom of it, and it spans the outer membrane. Transferrin binding protein B flops away at the surface of the bacteria and reaches out and captures on the outside of the polysaccharide in the case of the capsule, in the case of meningococci, but on the surface of the gonococci as well, captures transferrin and rotates it and docks it down so that the opening that allows it to release iron now is continuous with the opening of the inside of the beta barrel of the transferrin binding protein A. And this allows iron to be taken up into the cell. And it can use this very effectively. If we immunize with TBPB, and this is the one that's highly surface exposed, it has to flap at the surface and catch big proteins like transferrin, which is large like an immunoglobulin would be, for example. If we look at sepsis challenge, as I showed you before, this is looking at meningococcal survival. Bexsero will protect against meningococcal invasive disease in mice. If we immunize with TBPB, it completely protects against invasive disease by the meningococci. And this is showing nasal colonization with immunization with Bexsero and with TBPB. And I use the meningococci here instead of the gonococci to make the point that this reflects what we see in humans. Bexsero doesn't reduce colonization of the nasopharynx or nasal passages when immunized, but TBPB does. And we think part of this, and I'll come back to this later, might be the fact that it can both kill the bacteria, target the bacteria, but also starve it of iron. And I'm going to speed up, jump over these next little bit quickly so I can wrap up. But one of the important things that we had going for us, we're studying a human-specific pathogen, Neisseria gonorrhoeae or meningococci, in humanized animals. But other bacteria that are specific for other animals also have transferrin receptors. So there's pathogens of cattle that only bind bovine transferrin. There's pathogens of pig that only bind pig pathogens. And so we were, this is really the work of Tony Shrivers in the University of Calgary. They went ahead and tested whether they could use transferrin binding protein B as a vaccine using these surrogate models. So TBPB from a pig pathogen in pigs, for example. And what they found was that a binding defective mutant was a much more effective antigen or much more effective vaccine than the wild type antigen. And I'll come back to that. But by making a point mutation within the binding interface so that it repels transferrin instead of binding it, this binding defective mutant was much more effective. And that's shown here. This is an invasive infection with piglets. The piglets rapidly succumb to infection. There's a vaccine that's commercially available, Bacillus claser. It protects some of the animals, both the length of survival and the number of animals. There were a few animals that survived. If Tony's group used wild type TBPB, they got better protection than the commercial. But if they used a single point mutant, they got complete protection of the piglets. And in fact, it's not just survival, but they had very little symptoms of infection. And so this is a really important difference, a very exciting and important difference between transferrin, just a single amino acid change, and getting this much more effective protection. Importantly, these two immunogens are no different in a mouse or a rabbit. They look exactly, everything about the response to them is exactly the same. So this only happens when the native TBPB combine the transferrin that's in its host. Also important, in a different study, they showed that these were safe in pregnant sows and that they could transmit antibody to the piglets that would protect them from being colonized by this pathogen. So we wondered whether we could use this approach against the gonococci, and we just literally last week put this on bioRxiv, so if you're interested more, you can follow up. Trevor Moray's group at the University of Toronto selected a variety of different mutants to see if they could interfere with binding between transferrin and transferrin binding protein B from gonococci. They found their favorite mutant that has absolutely no binding affinity at all. They've shown by a variety of biophysical methods that this protein is completely stable. It doesn't look any different by whatever approach they look from the wild type protein. But it's actually, when we put them in the transferrin expressing mouse, the HTF transgenic animals, it's much more protective. And so this is looking at the amount of antibody against the wild type, the NGO1, or the mutant. TBPB, they're both the same. We don't see a different amount of antibody response. But when we look at colonization of the animals with gonococci, you can see that they clear much more effectively when we use the D209 mutant. And we think this is because of the fact that the antibody that's produced is different. It's targeting different regions of the protein. So this is immunizing a wild type mouse and looking at the ability of the antisera to block human transferrin binding. So this is looking at gonococcal binding to human transferrin. In sera from the adjuvant alone animals, it doesn't block binding. In sera from both the wild type and mutant, in the wild type, from when we immunize the wild type mouse, it's no different. They both block infection. But if we immunize a human transferrin transgenic animal, now you can see the mutant blocks equally to the wild type animal, but the gonococcal antigen that binds transferrin no longer does. And so we think this is due to an interaction perhaps between gonococcal TBPB and the transferrin that's in the tissues when we immunize it. The other important finding from this by Jamie Fagan in our group is that when we immunize with a wild type transferrin binding protein B and a human transferrin expressing animal, we now see an immune response against the human transferrin, which we didn't see in animals alone or alum immunized animals, and we don't see when we use the mutant transferrin. This effect goes away. So there's two effects. When we make a protein mutant, we get much better protection. This protection seems to be from blocking iron acquisition in part, and we don't get an autoimmune, a self-response against the host protein. And the last point here, and I won't spend any time with it too much, is that one of the challenges that we had is that there is a diversity of sequence variants across the different TBPBs. And so this is immunizing with five different gonococcal TBPBs from around the phylogenetic tree. And you can see that when we're looking at a number of different gonococcal strains shown on the left-hand side, if we immunize with a TBPB, it tends to confer reactivity against those gonococcal isolates that have a TBPB closely related to it. And these are each column within the broken down columns, each column is pre-immune serum and after 1, 2, and 3 doses. What we found is that if we combine two of these, TBP2 and TBP5, we see broad cross-reactivity, and this is equal to what we see when we get all five. And so by combining these two different immunogens, we get really broad spectrum coverage. And this not only just reacts with TBPB, it blocks transferrin binding by the bacteria. And it elicits a serum bactericidal activity. I'm sorry, I'm going quickly. I'm trying to wrap up. And very gratifyingly, when we combine these two mutants and immunize the animals, regardless of what the second version that we use, that we had two different variants, we get much better protection against gonococcal infection in HTF expressing mice. So I'm going to end there. This last part of the project really was about the vaccine, and I think we think this targeting this protein is an effective strategy against the gonococci because it's essential for the bacteria to get iron. Iron's used in DNA replication and oxygen metabolism and others. And we think the antibody response when we make a mutant allows us to both starve and kill the bacteria to effectively combat it. The vaccine work, the last two sections, looking at correlates of protection and the vaccine work is really a collaboration between my group, Tony Shriver's group at the University of Calgary, and Trevor Murray's group at the University of Toronto in biochemistry. And a large part of this funding is from National Institute of Health in the US, but also some from the CIHR as well. And I'll end there. Thank you so much.
Thank you, Dr. Grail, that was an excellent presentation. We have a question on the Q&As. So Rebecca Villa is asking us a three-part question. With the meningococcal vaccine, any idea how long the vaccine-induced protection against gonorrhea would last? And also wondering if there are any animal models for nasal Neisseria gonorrhea inoculation, and could you recommend papers or other sources where she could learn more about the model? And what do you think needs to be done to elicit vaccine-induced protection in the oropharynx in humans?
Yeah, those are all big questions. Everything that we know, I guess, seems to suggest that the protection by the meningococcal vaccines probably lasts about 2 to 3 years, I think. And so my group is immunized, for example, and we've started to boost because we work with meningococci and gonococci, and we started to boost them. And I think the durability of protection really in infants and adults that get it is probably around there. What's interesting is when young children get 4C-MenB, for example, the vaccine, they consistently have a very robust response. But when adults get it, that's not always the case. Some individuals don't respond very well at all. And so we think that this probably is something to do with this idea of original antigenic sin, where somehow they've seen either meningococci, a commensal Neisseria, or gonococci before, and it's suppressing the response against other antigens in the vaccine. With respect to the nasal infection model or the pharyngeal infection model for Neisseria gonorrhea, we haven't published this yet. So what I've talked about, I don't know if I alluded to it today. We do have this established in the lab now, and I would refer you to me, who would put you in touch with Jamie Fagan, who's a senior research associate in my group, who's developed a model, and she can colonize the pharynx with of CCAM humanized mice with a variety of different gonococcal isolates. And we think this is promoted also when we bring in the other factors such as human transferrin. We have published work on meningococcal nasopharyngeal colonization, including a new paper in bioarchives that shows the impact of transferrin and lactoferrin along with CCAM1 in colonization. And then what do I think needs to elicit protection in the oropharynx is really difficult. We've been very fortunate. Again, I use gonococci and meningococci to inform each other all the time. With meningococci, when they target the polysaccharide capsule, it tends to be that it can confer sterilizing immunity. So a serogroup A vaccine that's against the polysaccharide of meningococci tends to prevent any colonization in that population by serogroup A expressing meningococci. The group B vaccines obviously don't do that. They have no impact at all on colonization. There might be a hint against some strains, but really not anything significant. And so I think it might be a combination of two things. One, the density of epitopes on the surface might explain the polysaccharide versus a lot of the protein antigens, which are much lower density. And we've studied this using Bexsero against meningococci in the past, and that some strains in our CCAM expressing mice, some strains don't get protected if they only match one of the 4 antigens within Bexsero. So if they have a matching factor H binding protein or others, they don't get protection if it's only one. But if they match 2 of them, then we get protection. And so this, to me, suggests that you probably need increased antibody binding, so increased epitope density on the bacterial surface. And I think the other way to do this is to target an essential function, as I talked about with transferrin binding protein B today.
I just have a question. So a lot of these studies are done like looking at humoral response. Did you like ever consider looking at a Th1 response and like, because I feel like a Th1 Th2 balance might be a better approach than just focusing on antibody alone.
Yeah, so this study that we just published in MSphere looking at 4C-MenB and the adaptive responses to it and to try and identify correlative protection. I think there's, I don't know what there is, 17 supplementary figures, as well as the figures in the paper, because we really tried to look at everything we could. We looked at 44 different cytokines in the genital tract and systemically. We looked at lymphocyte responses in spleen after immunization and looking at not just B cells versus T cells, but breaking them down into Th1 versus Th2 type cytokine response and lymphocyte responses in the spleen and in the genital tract after infection. And really nothing shows, and there's a statistician on that paper who did just an incredible amount of work to try and combine different factors together. And there might be a little bit of a signal if there's about 13 different things that match or something, but really there's no evidence of what would be obvious. Mike Russell's group has done work where if they promote a Th1 response in the lower genital tract of mice during gonococcal infection, and so they did this by colonizing the animals with gonococci in the lower genital tract and then adding these IL-12 microspheres, or by introducing outer membrane vesicles into the vagina along with IL-12 microspheres. So promoting a really strong Th1 response, then they tend to get protection in the animals. And so it might be something like just simply skewing the type of response that occurs naturally to infection. And what's interesting in that regard is that the response to gonococci doesn't seem to be, we don't get a good adaptive response. It almost seems like it's a low-level inflammation that is maintained almost to prevent an adaptive response from happening. And so he believes adding the IL-12 overwhelms this and really drives this response from a Th17, kind of an inflammatory neutrophil type response to a Th1 response that confers protection. So there's hints that the part of the problem might be that the inductive site, the genital tract or the throat, is not a good place for it. And if we can skew the way that the bacteria are recognized, it would help us. And then the trick is to try and figure out how a vaccine can do that same thing.
Right, because I was also wondering because gonorrhea naturally is a induce a mucosal like infection. So would it be better to promote or like encourage immune response in the mucosal surfaces? Like if it is a Th2 response we want, like do we want IgA more than IgG? And how do we like
Yeah,
promote that type of response?
yeah, that's an interesting question. With with reinfection, again, we use the meningococcal model with reinfection in the nasopharynx, so nasal passages. You get a really localized IgA response and very little IgG systemically, and we do get protection with that. As far as we know, reinfecting the lower genital tract in the mice doesn't confer protection.
All right, I think we are at 1 o'clock. Thank you everyone for your time, and I would like to thank Dr. Scott Gray Owen very much for his time today. It was an excellent seminar. We would like to remind everybody that we don't have a seminar in December, but we have an upcoming seminar on January 29th by Dr. Vanessa Allen. So everyone is welcome to join our next webinar. Again, thank you so much, Dr. Gray Owen.
Thanks so much for having me. Thanks, everybody.